Okay, I'm going to talk today. I'll give a general overview about microplastic pollution, talk about some of the issues and perspectives related to methods. So I'll start by introducing the contaminant, and I'm going to talk about its multidimensionality and how it's unique from other types of contaminants that we often tend to think about, like chemical contaminants. Then I'll talk a bit about the methodological developments and challenges, things we need to think about, share some of the things we've been working on in our lab, and then some of the things I think we should think about as we move forward. So first of all, let me just introduce microplastics. So since I've been researching this contaminant, which has been since about 2007 or 2008, we've learned a lot. And we've learned that microplastics, like other persistent organic pollutants, are incredibly persistent, they're ubiquitous, and they can be toxic. They're not a persistent organic pollutant or haven't been designated as this, but they certainly have some similarities. They also have some differences. So before I talk to you about some of those differences, I want to start by just talking about how pervasive this contaminant has become. So plastics are entering the environment at an astounding rate, and they really become an important part of the physical environment in ecosystems worldwide. Researchers have found that they're cycling in the water cycle and the global dust cycle, through food webs, and also in the carbon cycle. But this contaminant is also unique, and I tend to think about it as a diverse and multidimensional contaminant suite. And what do I mean by multidimensional? Well, it's a particle. It in of itself is obviously made of chemicals, but they're physical particles. They're made out of many different types of polymers. That includes a suite of many different types of additives. They come in different shapes, from fibers to fragments to spheres. Those different shapes may impact their fate or maybe their toxicity on animals. They also come in different sizes, and similarly, those different sizes may affect their fate and their toxicity. The other thing about microplastics is that instead of having a half-life where they degrade and go away, they tend to break up into more and more smaller pieces. And because there's so many different types of these materials out in the environment, it's no surprise that they come from many, many different sources, which includes waste. It also includes fishing gear and other maritime sources. But also for microplastic, there are unique sources that are different or that are separate from plastic waste and maritime debris, and that's pre-production plastic pellets, the wear and tear from fibers and textiles, and microbeads in personal care products. They enter the environment from these sources, but often through pathways, which are important, especially when we're thinking about mitigation. So they come in water currents with rivers and also surface currents. They come in via the wind and atmospheric cycles, atmospheric deposition as well. They can also come in direct input to the ocean from wastewater running off of the land or treated effluent. And then once they're in the environment, their multidimensionality can affect their fate. So we tend to have ideas and knowledge, often from first principles, about how persistent organic pollutants cycle around the world, how they cycle in the atmosphere, and how they cycle in the water. For microplastics, however, there may be differences, and some of those differences are likely due to that multidimensionality. And for one example is that they're not volatile or semi-volatile like some of these persistent organic pollutants. Again, they're physical particles. In addition, this multidimensionality has to be considered when we think about risk. We know that microplastics can cause toxicity to animals. There's a few effect mechanisms that have been studied more than others. As we think about the concentration that may lead to risk thresholds to identify policy decisions, we have to think about, do we report it in count of particles per volume of water? Do we report it in mass? And does it need to vary by all of these multidimensional properties like size, shape, polymer type, and additive chemistry? All of these questions at their core, to be able to really answer them and understand them, are relevant to methods. We have to be able to analyze this stuff in the environment, and we have to be able to understand certain physical characteristics and properties in order to do so. So there are different methods that have been proposed or that are being used around the world. Those include measuring the mass concentration of microplastics through gas chromatography, mass spectrometry, often with pyrolysis unit at the top, or it's by particle number concentration, often starting with microscopy, but sometimes going straight to spectroscopy, where you get things like the count and the size distribution and the polymer type, and sometimes the shape and the color, all of which can be important for informing sources, understanding the fate, and of course, thinking about the effects. So now I'm going to talk a bit about the methods and the things that we have to consider when we think about methods. It won't be a laundry list, I won't have them all, but I'm going to mention some of the ones I think are really important and share some of the work we've been doing in our lab in order to address these. Now, I never considered myself a methods researcher or a methods scientist. It was, I guess, never my goal, but it has been a lot of my work, and that is because, as I said, in order to answer these questions, you need methods. And so we've probably spent a fair chunk of our time in our laboratory thinking about this stuff. And when we think about methods, we want to think about sampling, all the way to sample extraction, sample analysis, and all the way through to reporting. So I'll go through each one, some of the things to consider, and then share a bit about what we've done related to each. First of all, when it comes to sampling, we really have to always, I think when we think about any method, think about the objective and our question going into our research. Then we need to think about the sample volume. How much volume of water or sediment or animal do we need to sample in order to get a representative sample from what is in nature? What is the sampling method that we should use? Will it be bulk water sampling filtered through a small mesh, or will it be a Manta trawl, which has a larger mesh on the back, sometimes up to 1 millimeter, often around 330 microns? When we sample, how many replicates will we take? Will we take one large toe in one location, or will we duplicate our effort at each location to understand variability? And as we sample, will we take field blanks to be able to account for that procedural contamination? And that field blank can also come into the lab, acting as a lab blank. So we've explored some of these things through a couple different projects. I'll share with you some of what we've discovered or trends we've seen through a project with the San Francisco Estuary Institute, quantifying and characterizing microplastics in the San Francisco Bay. In this study, we took Manta trawl samples as well as bulk water samples from the exact same location at the same time from a sailboat within the San Francisco Bay. What we're looking at here in this figure is the particles per liter, so the concentration of microplastics on the y-axis. And each one of these are duplicate samples taken back to back. So not only were these samples taken at the same time, but there were duplicates taken back to back for each type of sample. And so the first thing I want you to notice is that you have a much higher concentration of microplastics in the Grad sample than the Manta, but we'll talk about that more in the next slide. Here, I want you to focus on the variability between duplicates. You see much smaller variability with a Manta sample when you have a whole lot of water, and you see much greater variability with a low volume Grad sample. Now these are very low, 1 liter. I don't recommend sampling with this low of a volume, but if you do, absolutely take field duplicates to be able to understand the variability. So the lower the volume, the more important those duplicates become. As I said earlier, you can see here that the concentration of microparticles is much higher in a Grad sample than in the Manta. This is for two reasons. One is when you're using a smaller mesh size on a Grad sample, you're going to have more particles because you're capturing more of the small stuff. The other reason, though, is because I'm not sure what the reason is, but there's a lot of work done across the field where people have been taking Grad samples and they've been taking Manta samples. And when you have a smaller volume, they're finding that smaller volume samples tend to have a higher concentration. Now, this may be because simply of the mesh size, but it's something to further be explored. When you have a smaller volume Grad sample next to a Manta, we tend to find that the Manta is more representative of the shapes and morphologies and types of plastics we see in the environment. However, I've also seen we've been doing a systematic review and meta-analysis comparing these two methods, and with a high enough volume for a Grad sample, these can be equal. But you absolutely, I think if you're taking Grad samples, the take-home message is make sure you take enough volume. Okay, now let's think about things to consider when it comes to sample extraction. Now, of course, the first thing is going to be the extraction method, which is going to vary, of course, by the matrix that you're sampling in. And that may tell you whether you do a density separation, some sort of just a simple filtration, maybe for clean drinking water, or whether it's digestion of biota, fish tissue, or even a really dirty Manta sample with a peroxide oxidation method. No matter whatever method we use, the most important things we should think about is recovery and precision. How well are we recovering the particles we aim to recover in our sample? We should always have lab blanks, which if you've taken field blanks can be carried over. This is to account for procedural contamination. I think it's really important that those blanks go through the entire process, same as your sample. I don't think putting a filter out in a lab is the solution, because that is a very different process than what the sample actually goes through. So I think similar to analytical chemistry, we should have lab blanks that mimic our sampling and sample prep. And then the next question is, do we subsample? We don't hear a lot of people talk about subsampling at the extraction phase. We hear a lot of people talking more about subsampling at the analytical chemistry phase, but I'll talk a little bit later why I think this phase could potentially be another place to consider doing it. But the examples that I'll show you here are going to mostly be around extraction. There have been a lot of different extraction methods that have been proposed. Some of them are quite novel. We have been guilty of this. For example, we have a manuscript that describes a study led by Brian Nguyen and Jelena Gerbic, where they magnetized the plastic using a solvent and iron nanoparticles and a magnet. They were able to extract microplastics from different types of samples, and this included larger than 1 millimeter plastics of many different polymer types, very small 10 to 20 micron plastics with decent recovery, and across a wide range of matrices. However, the reality is I'm not sure we're going to pick these types of novel methods if we're trying to harmonize around one method. And the most important thing is we need to think about recovery and we need to think about precision as we determine a best method for matrices, particularly as places around the world lead towards monitoring, which is what's happening right now, which I think is fantastic. So in our lab, the other thing we've been working on, because we're collaborating with the Southern California Coastal Water Research Project, SQURP, and the State Water Board to do a method evaluation study for the state of California, but really there are participants across the world. And for that, we had to send out samples that we spiked. We knew what were in them. Actually, Thermo Fisher is heavily involved in this project as well as one of the labs helping with this project. So we spiked samples, we know what's in them, we send them out, we send out an SOP. And we also did some training for people who are new, and we asked people how many years of experience they have in microplastics. One of the things we've learned is as you set up these extraction techniques in your lab, experience and training matters. So we've put our training materials on our website if people are curious to take a look. And this work has largely been led by Hannah DeFrond and Keenan Munot, who are researchers in my lab. So again, experience and training matters. As you choose your method with recovery and precision, it can be improved over time with experience and also training protocols that help keep subjectivity as much as possible out of the lab, right? There will always be a human dimension. The next thing to consider is sample analysis, right? Or chemical analysis. So once you have taken your sample, you've extracted your sample, you've potentially you counted your sample under a microscope, and now it's time to determine how many polymer types do you have within your sample. So here the first question is, what is the type of analysis that you will use? Will it be a spectroscopy using Raman or FTIR, or will it be mass spectrometry? Are you actually interested in the mass concentration of each different polymer type, or are you interested in the type of chemicals? And of course, mass spec can also be used particle by particle and sometimes does well with complex particles like tire dust. What polymer spectral libraries will you use? What's available in your library will help you better pin down the types of contaminants or microplastics in your sample. How do you present your sample to the instrument? That'll affect how you prepare your sample preparation. The limit of detection of your equipment. This should be reported in studies. It isn't always, and we've been guilty of that too. And then again, this question of subsampling. We see a lot of people across the literature subsampling either the particles from a sample to figure out what is in them, or subsampling from a filter. So I'll talk a bit about libraries, and then I'll talk about subsampling, because that's some of what we've been thinking about quite a bit in our lab. So first of all, when it comes to libraries, in our lab, we've developed SLOP and FLOP, which is the Spectral Library of Plastic Particles for ramen, SLOP ramen, and then FLOP is the FTIR Library of Plastic Particles. FLOP is coming soon. It's been led by Hannah DeFrond in my lab and in close collaboration again with Thermo Fisher. And SLOP is already published in the literature and freely available on our website. So our goal here is to make open access libraries that are loaded with spectra from plastic particles that look more like the plastic that we are commonly analyzing. So right now, often when you get an instrumentation, the library has pure particles, they are colorless, and they're kind of perfect. And we all know from the microplastics research that those microplastic samples aren't perfect. They come in a range of colors, they come in a range of sizes and shapes, they have different additive chemistries, and sometimes they've been weathered in nature. So we've created spectral libraries using both types of instrumentation that incorporate this diversity. So we include many different colors of each polymer type, we include different shapes, and we include both samples from the environment that we've taken in different studies, as well as pure, not pure, but particles off the shelf that have been broken down and used in this study. One of the things that we find with this is for SLOP is that we have a lot of particles for ramen that sometimes we can't figure out what polymer type they are. This is because the laser absorbs the dark color and we get a pretty messy spectrum. This is sometimes because of band overlay with the additives. This can just be because the hit quality index isn't good because it's not something recognizable in the pure polymer library. And so we took many of these particles, I think about 60 to 100 of them, and we reran them once we had SLOP and sloppy. Sloppy is just the environmental particles within our instrumentation, so set up on our ramen. And here we find that now we have less particles with this unknown signal, and we're more likely to be able to tell what they are. And we often get our own libraries popping up over the other libraries. Now for FTIR, this isn't out yet, we're still working on it, but in our lab, as we start to use FLOP, we're finding a similar thing, particularly for pesky particles like tire wear rubber. Okay, so now let's talk about subsampling. This is something I think about a lot. I look across the literature and I think a lot of us are subsampling without giving it a lot of thought of why we're doing it and are we sure that the sample, the method that we're using is representative. I think it's really important that we report on the objective of our subsampling, and then we also report on how we know that it is providing us the representative answer that we are trying to get. So I think when we subsample, there are two reasons that I'm going to propose. One is that you're just trying to understand how many of the particles that you picked via microscopy are actually anthropogenic and plastic versus natural particles. So how accurate is your picking under ramen? For this, you might want to be producing some sort of pie chart here where you can tell a reader the percent that is not plastic. And maybe this is to extrapolate to a sample. In order to do that, to ask this question, how many particles do you need to take from every sample if this is your objective? We got samples from around the world, from people who had collected all of the particles and who had analyzed via spectroscopy every single particle. We then used computer simulation to basically pick particles over and over again, continue doing many replicates, and to see how many particles do you have to take to get under a 20% error in building a pie chart like this from the true sample. And what you see here on these graphs that I'm going to show you is the summed error. So we're trying to get under this dotted line and the number of particles that you have to chemically characterize in order to be below a 20% error. We also ask the question, when you subsample, do you do it randomly, any particle that is in your sample, or do you do it by like choosing 5 of each color, for example, or 5 of each shape, or by color category, so 5 of each blue fiber, 5 of each black fragment, etc. We found actually that random is just as good as anything else, if not better, which is great. That means you don't have to worry so much about color and morphology. And so if you don't care about it in your sample, it doesn't matter for this purpose. We also found that here you can sample from just about any of the samples we looked at. 25 particles was the, like to be able to get what you wanted in every single sample, 25 particles from every sample would get you there. here's my second reason why I think people might be subsampling. This one's a little bit tough. I think this is if you're truly trying to capture the diversity of polymer types present in your sample. So now I'm not just interested in whether it's plastic or not. I'm actually interested in the types of polymers in my sample. So here, we want to capture the diversity within our sample. But one of the things we found is that it actually looks like we might not even be sampling enough to capture all of the particle types that are in the environment. And that's a different story, and we won't dig into that today. But in any case, we were thinking about it from a bit of a community or from a community ecology perspective, thinking about a rarefaction curve, looking at our samples and recognizing that as we sample from the environment, our curves weren't flattening at the top, and it was suggesting maybe we need more volume. But in any case, for this study, we worked off of the samples that we had, and basically we found the same thing. Random is just as good, if not better, than other types of sampling. However, now you have to sample a lot of particles in order to capture that diversity. People might not have the time to sample this many particles, so it's really important to think about, again, what is your objective for subsampling, and make sure that you're capturing that. This is work led by Hannah DeFron and also in collaboration with Anna O'Brien from our lab. Not published yet, but getting to that writing stage. Finally, when it comes to subsampling, we thought about subsampling from a filter. So Clara Tyson in our lab led this work. And the reason we were doing this is because we were considering subsampling from a filter, and we looked across the literature and it looked like people were doing it, but it didn't look like there was any clear role of how they were doing it representatively. So we tried it out. We filtered many different samples. We tried many different ways to subsample. Do you randomly put squares on a filter? Do you draw rings around it and do a square in each ring? Do you do pizza pies? We never got anything that was representative of that subsample if you scaled up. Nothing was ever below a 20% coefficient of variability. And what we found, though, is that you always get this kind of sunburst pattern, and that you have a lot of particles in the middle and less as you go out. And you can see this from the filters here, more particles in the middle and less as you go out. So this suggests that you have some sort of pattern that maybe you can work off of. But because this seems to be tricky, it also suggests maybe we should subsample from the liquid, homogenizing your liquid and subsampling from that and putting less particles on the filter so you don't have to subsample your filter. And I think that's our recommendation for now. Other things to consider that I won't go into great detail about are QAQC. There have been papers written. There was a special issue in applied spectroscopy that came out last year, also part of the Southern California Coastal Water Research Project program on method evaluation study. We published a special issue. We had a couple papers about QAQC and the things you need to consider, reporting on lab contamination, reporting on recovery, etc. It's really important to think about our units of measurement. When we think about reporting, we need to report what they are, and we also want to make sure that they're kind of transferable across the literature so that people can do meta-analyses. And I personally think it's important to make our data open access. We've sort of been in the habit of throwing our CSV files and Excel sheets and data sheets online as we publish. And I think this is really useful as we try to synthesize information around the world to really try to understand this contaminant. Okay, and the final thing I want to say is just a few thoughts on the things I think we should be thinking about as we move forward. In order to answer some of these questions in terms of the multidimensionality of plastics, we should be working towards more harmonized protocols, not necessarily a standard protocol that everyone uses, although that may be desired in certain states and provinces or countries as monitoring programs are put in place. But harmonized protocols are really important, that have clear reporting guidelines. What are the types of things we want to see in a manuscript or in a report in order to be able to trust the data? Recovery, for example, could be one of them. Making sure you report the size of your material, your hit quality index, whatever we decide is important. We need better streamlined and high throughput methods. This is where a lot of the manufacturing companies really come in. They've been making huge leaps and bounds over the years. And that's really helped, right? Help us improve our accuracy and our precision, but also the amount of time it takes to process a sample. We've thought a lot about whether we need standardized morphology and color keys for particle characterization. I think color and shape are important to help us understand the source, but it's subjective. And then, of course, clear QAQC guidelines, as I mentioned before. We need clear, clean lab techniques that are expected of us. We should think about whether we require matrix spikes or at least some sort of way to report on your recovery within your extraction protocol in your lab. Blank sampling, I think, is really important, whether it's field blanks and lab blanks or field blanks that carry into lab blanks. And then how do we correct? Are we blank correcting or are we simply just reporting? And what's our limit of detection? Should it be based on the size of the particle, the concentration of the particle, or the amount above the blanks, or a combination of all? Of course, it's going to matter whether you're using mass spectrometry or spectroscopy, because some of these things are going to be relevant to one and not the other. But I think these things are all really important. And while we can look to the field of analytical chemistry to guide us, and we should, we also have to remember the differences. So I'm going to leave you again with this slide one more time to just remind you that this is it's a diverse contaminant, it's a multidimensional contaminant, and not all microplastics are the same. And as we develop our field moving forward, we can't forget that, that this is the case. And make sure that we move forward accordingly. And with that, I want to thank you. I also want to thank Thermo Fisher for inviting me to this workshop and collaborating with us over the years.